It's a good thing to get clear at this point. So we're going to look at two meioses going from left to right. And again, as we discussed in class, this looks like male meiosis. So we've got 4 equally sized gametes at the end. So we're going to track, and this is post-synthesis. So synthesis happened, time moving from left to right. Synthesis already happened to the point where we get to a cell that has, let's see, how do I want to do this? Colors will represent chromosomes. So we have 2 paternal versions of the red chromosome and 2 maternal versions after synthesis. And then blue chromosome, smaller chromosome. Also, you've got 2 copies from dad and 2 copies from mom. Paternal, paternal, maternal, maternal. Okay. So how am I going to arrange? Here's the first division. How do I arrange the red chromosomes in those 2 daughter cells? Let's just start with the easy question. How many red sticks am I going to draw in each of those cells? You start with a 4, so I'm going to divide the red sticks, the chromosomes, in half. So each of those we're going to get 2. And the same is going to be true for the blue chromosomes, hopefully, unless you're one of the oocytes that we were just looking at in class. So far, so good? Yes. So we started with 4 reds and 4 blues, and we now still, at this step, have 4 reds and 4 blues, and second, totally separate meiosis process, 4 red chromosomes and 4 blue chromosomes. So let's add in the nomenclature. So the 2 red chromosomes here in the top, what letters are those? They had letters here, so they're either P's or M's. So you want to do P's? Okay. So we'll call that P. And that must mean then that the red ones on the bottom are the M's. Okay, so now what? For the blue, same thing. Yeah.
M's?
On the top?
Yes.
Okay. And that means
P's.
like that. Yep.
Okay.
And then cell division happens. So we wind up with a red chromosome in each haploid gamete and a blue chromosome in each haploid gamete, and apparently a little blue dot too, but that's, I don't know, chromosome broke maybe. So then those red ones on the top are both P's, and the red ones on the bottom are both M's, just filling in from left to right. The identities of the chromosomes haven't changed. So that's how you get those haplotypes. Red chromosome from dad, blue chromosome from mom. You get 2 of those. You get 2 gametes of a different haplotype. Red chromosome from mom and blue from dad. So the point about that video for meiosis part B, the orientation of different tetrads of meiosis, is what else could be happening here? So there could have been an alternative arrangement, but now I cannot erase that text anymore, so I'm just going to draw over it. Think about these tetrads as they're just floating in the middle of the cell, and the nucleus, really. So instead of writing them in the same orientation, PP, MM, from left to right, this could have also been MM, P, P. There's no genetic difference between that or the way it was originally written. It's just that the 4 chromosomes are just rotated inside the nucleus when they're lining up before cell division. But that makes all the world of difference in what happens in the outcome of meiosis. So if that means that, let's see, let's assume we keep these in the same orientation. We put the P's, the red P's on top, and the red M's on the bottom. Then the other orientation of these is you put the blue P's go with the red P's, and the blue M's go with the red M's. So that's like... Oh, I did draw it right the first time, didn't I? Crap! Yeah, so that's those segregating in one direction with those into one cell. And those versions of two different chromosomes segregating together into the top cell. So you've got P's with P's and M's with M's, and when you wind up at the end, that's the haplotypes of the gametes. You have the two paternal versions of each chromosome in half the gametes, and you have the two maternal versions in the other half of the gametes. So then you've got 4 different haplotypes. And the assumption in meiosis is that we've just looked at 2, right? We've looked at one cell here going through meiosis, and we've looked at a second cell. Normally, organisms do meiosis hundreds of times. It depends on the sex, of course, too, but hundreds of times, thousands of times, millions of times during their lifetime. So the argument is, on average, every time you go through meiosis, one or the other of these will happen. So if you do meiosis 100 times, you'll get an equal number of those haplotypes, those haplotypes, those haplotypes, and those haplotypes. And that produces a 1-to-1-to-1-to-1 ratio of haplotypes in the gametes.
Those the only 2 options you have?
Are there more?
Yeah.
No, that's my question back to you. Are there more different arrangements? So if you have the red, and this is Mendel's second law, independent assortment of alleles, meaning that different chromosomes do whatever the hell they want regardless of what the other chromosomes are doing during meiosis. So look at all of the cells of these 8 that are on this screen. Look at all the cells that have red P chromosomes. There's 4 of them. There's one there, there's one there, there's one there, and there's one there. So it's the top 2 in both of these examples. So you look at the blue chromosome. This is the independence. Half of those that have the red P chromosome, the P version of the red chromosome, half of them have the M version of blue, that's those 2, and the other half have the P version of blue.
Yeah, I suppose if you did it a different way, you still end up with the same
ratio. So, yeah, so when you look exactly, if you look from the perspective of one chromosome, like the red P chromosome, there are only 2 different ways that you can be combined with 2 alleles of another chromosome, half with one and half with the other. And we won't get more complicated than this, because then you could start thinking about, well, crap, humans have 23 chromosomes. So how many different possible combinations of haplotypes are there when you say, okay, if we looked at human chromosome 1, what's happening at human chromosome 2? So there are 2 ways. This is exponential. That's the punchline. There are 2 ways to arrange 2 alleles of one chromosome. Half of the gametes get the paternal version, half get the maternal version. If you add a second chromosome, you've got 2 to the second power. You've got 4 ways to arrange the chromosomes. You start looking at 3 chromosomes and how many different possible combinations, then there are 8. And 16 and 32 and 64 and 128 and 256, 512 and so forth. So if you do 2 to the 23rd power, that's how many different, that's why we don't do it. Say, draw out all of the possible haplotype combinations you get from human meiosis. No.
Yeah, how about no?
Yeah, definitely no.
So, how long would that take you?
A while. So it literally is all about how the tetras line up during meiosis. So for chromosome 1 and chromosome 2, totally different example, but trying to repair the damage I did on the previous slide. If that's how the tetrads line up at meiosis, we know that the sister chromatids stick together, that is the two versions, one homolog and its duplicate that just got synthesized synthesis, stick together more tightly. So they're the ones that are going to separate at meiosis 1. So that's one way to arrange these tetrads. The only other way to do it, in red, is for that group of 4 chromosomes, literally inside the nucleus, to be rotated 180 degrees, so that you get the paternals on the other side of the mitotic or the metaphase spindle, and so then you get that happening with chromosome 2. So in the first case, you get paternal chromosome 1s with paternal chromosome 2s. And in the second case, you would get paternal chromosome 1s with maternal versions of chromosome 2. And those are the only two choices.
Okay.
But that's the entire basis of sexual reproduction in a nutshell, it's this process. It's the fact that you take some of mom's chromosomes and some of dad's chromosomes, and you make a big mess of them all. Take random combinations of them, you put them in your gametes, and then you generate new combinations of genotypes, new haplotypes. This is the question from the meiosis video to talk about in class today. So you've got the genotype A1 over A2, that's one chromosome, and then B1 over B1. So post-synthesis, as we looked at in class, then there are 2 copies of chromosome A version 1 and 2 copies of chromosome A version 2. And then all of the chromosome B's are the same haplotype or the same allele. It's all B1. So here it doesn't matter what the orientation of that second tetrad is, because they're all the same genotype, all the same haplotype. So it doesn't matter how those ones segregate or get split up into gametes. The only thing that matters is that half of the gametes are going to get allele A1 and the other half are going to get allele A2. They are all going to be combined together with a B1, because that's the only choice we have for that chromosome, is that you get B1, like it or not.
Wait, so A1 over A2, that means you have 4?
No, that's after synthesis.
So before synthesis, what do you have?
Pre-synthesis, you've got one copy of chromosome A version 1 and one copy of chromosome A version 2.
Okay.
That's what that genotype literally means. It's one copy of the chromosome has 1. The slash means now we look at a different chromosome.
That's
and your other chromosome...
Wasn't semicolon...
Semicolon means different chromosome. So this is chromosome A. You've got 2 copies because you're diploid. You've got 2 copies of chromosome A, and you've got 2 copies of chromosome B.
Oh, so semicolon would be...
Semicolon means we're looking at a different chromosome.
Chromosome B.
Yeah, chromosome A versus chromosome B. And this is where the nomenclature, as you realize, gets tricky, because when I say chromosome, you don't know if I'm talking about homolog, which is A versus B, or if I'm talking about here's a chromosome, here's a chromosome. And that's where the term homolog and the term sister chromatids come in handy, because I could refer to those as sister chromatids or homologs. And then if I say one of those 2 words, you know I'm talking about that chromosome versus that chromosome, as opposed to chromosome A versus a totally different chromosome that has different genes, different banding pattern, different size, different centromere position, literally different chromosome.
And if we do get confused with that, because I have a feeling it's going to be on your test, we can ask you, right?
You can ask me during a test, but...
Because the wording was the issue
yeah,
last time?
we'll see. It depends on the circumstance, but yes, you can always ask. The worst thing that can happen is I'll say no. I'm not going to tell you. If that's the thing I'm trying to test you on, then I won't tell you. If the intention was that I just want to be clear, then yeah, of course I would tell you, and I would also tell everybody else. Usually during an exam, if somebody brings up something that's clearly misleading or wrong or unclear, then usually I'll stop everybody for a second and try to clarify. Yeah.